Welcome to the Enzyme Tales. Enzymes are biological catalysts that speed up chemical reactions in living organisms. Today we have a special story about sucrase, whose primary function is to catalyze the hydrolysis of sucrose into its two monosaccharide components, glucose and fructose. Sucrase is essential for digestion because the body can absorb monosaccharides but not disaccharides like sucrose. In our story, sucrase is the villain that goes lurking for disaccharides to break them apart, specifically all the sucrose molecules. A sucrase molecule is a type of disaccharide composed of two simple sugars, glucose and fructose. Sucrose is commonly known as a table sugar and is widely used as a sweetener in food. Monosaccharides are the simplest form of carbohydrates and serve as the basic building blocks for more complex carbohydrates such as disaccharides like sucrose, oligosaccharides, and polysaccharides like starch and cellulose. Sucrase is never alone when it attacks. The active site contains amino acid residues that participate in catalysis, typically including glutamate or aspartate, acting as proton donors or acceptors. Other polar amino acids stabilize the substrate through hydrogen bonds or electrostatic interactions. Lastly, a water molecule acts as the nucleophile, as it has a lone pair of electrons on oxygen. The glycosidic bond between glucose and fructose is the electrophilic target. The catalytic amino acid, typically glutamate or aspartate, assists by deprotonating the water molecule, turning it into a more reactive hydroxide ion, which is a much stronger nucleophile than water. This story takes place in the land of the small intestine, specifically in the town of the Bruges border membrane of the intestinal epithelial cells. In this town, thousands of molecules live and engage in different reactions every day. Some of them start with the smallest of molecules. Let's take a look inside the monosaccharide daycare and see what's going on. Inside, there are a bunch of little monosaccharides waiting to find friends to react with. Today, two of them have found each other. One is an alpha glucose. The other one is a beta fructose. You can tell by the position of their OH group found in the first carbon.
Hi, my name is Glucose.
Hi, I am fructose.
Do you want to be best friends?
Yes, I would love to. But how do we become best friends?
That's easy. We just have to create super handshake. Call it glycosidic linkage.
Yes, that's perfect. And poof! As if by the magical art of biochemistry, the two monomers became a sucrose molecule. Their glycosidic linkage is a covalent bond formed during a condensation reaction, also called dehydration synthesis, where a water molecule is released. As soon as the reaction happened, the polysaccharide teacher came to congratulate them, but also to warn them. Congratulations, you are now sucrose. Your glycosidic linkage has turned you into a disaccharide. As a sucrose, you have to be very careful. Always remember, you can never go to the epithelial forest, where the microvilli trees are. There lurks the SUCRASE Enzyme. You must never cross their path. Otherwise, your friendship will be broken forever. And it was true. Lurking in the forest of the microvilli was the sucrase enzyme, ready to catalyze the hydrolysis of any sucrose molecule coming its way. The little sucrose molecule got upset after hearing such news, but the molecule was newly synthesized and naive and thought to itself,
Maybe our friendship is so strong we might be able to defeat sucrase.
Yes, let's do it! And so the molecule entered the scary forest in a quest to defend all the other sucrose molecules that have yet to come. However, it didn't know that the moment it stepped foot on the forest, there was an enzyme ready to act. Are we going on the right way?
I'm not sure. All I can see are trees.
What's that sound?
I don't know, but I'm scared. Hello, little disaccharide. What is a nice molecule like you doing here? I am here to defeat the sucrase enzyme.
Yes, that's right.
Have you seen that evil vermin lurking around? Not particularly tonight. Maybe they'll come around tomorrow. It's very late. Maybe you guys should head back home. Although my house is very close. You can rest there until tomorrow if you want.
The sucrose was scared to go with this new fella, but the thought of spending the night alone in the forest was even worse. The trail was confusing, and surely tomorrow it would be easier to head back to town. So little Sucrose finally accepted to go with the stranger. Okay, we'll go, but just for the night.
Perfect. You don't have to worry anymore. I'll keep you safe. Mwahaha! They have fallen right into my trap.
And so the little sucrose molecule followed the stranger into the depths of the forest. As they were walking, the stranger grabbed the little molecule and the reaction started. Sucrose was bound tightly to the active site of sucrase. The catalytic amino acids in the active site facilitate proton transfers that stabilize charge intermediates and transition states. So first, it was the nucleophile activation. Water is deprotonated and its electrons form a hydroxide ion. Second is the nucleophilic attack. Electrons from the hydroxide ion attack the electrophilic anomeric carbon in the glycosidic bond, leading to cleavage of the bond. Third, protonation of leaving group. Amino acid residues donate protons to stabilize the leaving group fructose by accepting the electrons left after bond cleavage. Transition state stabilization. 4. Enzyme active site residues stabilize the transition state by interacting with the shifting electron density. 5. Product formation. New bonds form, leading to glucose with an OH group attached and the release of fructose. Finally, the SUCRASE enzyme split the bodies of what once was sucrose. There, the bodies were absorbed through the intestinal lining into the bloodstream for energy production and metabolism. The little molecule was never seen again, but soon other sucrose molecules would come and meet their end, just as this one did. Ha 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 ha. And so it was the end. Remember, as food passes from the stomach to the small intestine, sucrose from foods such as table sugar fruits and processed foods is broken down by sucrase into glucose and fructose. Have a happy Halloween!